Microscopes are an important tool in the microbiology lab, so it is important that we know how to use and care for them. Cells and bacteria are very small. They're typically measured in micrometers. Now, 1 micrometer is 10 to the negative 6 meters, or 1/1 millionth of a meter. Viruses are even smaller. They're measured in nanometers. A nanometer is 10 to the minus 9 meters, or 1/1 billionth of a meter. So these are incredibly small things. The compound light microscope is what we use to enlarge cells for viewing. It's called compound because it has two magnifying lenses. It's called a light microscope because it uses light for viewing. This is the most commonly used scope in the laboratory. Now, two important parts of the microscope are the arm and the base. Microscopes used to come in two pieces, and the arm would come off of the base. When you carry the microscope, you always want to use both hands. One hand goes under the base, and the other hand holds the arm. Again, this is sort of a holdover from when the microscopes would come apart if you picked them up using only the arm. However, today's microscopes are pretty heavy, so using two hands is a good idea. Also, you want to carry the scope close to the body so that you have good control of it. The ocular or eyepiece is the part of the microscope you look through. The nose piece holds the objectives, and the nose piece will rotate so that you can move the objectives and change the magnifying power of the microscope. The stage is the area where you're going to put the slide, and the light source is here in the base. If you look closely at the base, you will find two knobs on the arm. The outer knob is the coarse adjustment knob. This is used for initial focusing. And then the inner knob is the fine adjustment knob. This is used for adjusting the focus when you have something more or less in focus. The stage is where you put your microscope slide. Underneath the stage is the iris diaphragm. This controls how much light gets to your eye, and the iris diaphragm lever is how you open and close the iris diaphragm. Hanging from the bottom of the stage are your stage adjustment knobs. This is how you're going to move the slide around on the stage. There are several ways to turn the microscope on. You just have to look at your microscope and find where the on switch is. Some microscopes also have a rheostat for changing the intensity of the light. Never use this as the way to control the light that gets to your eye. Always have this on full intensity and then use the iris diaphragm to control how much light gets to your eye. If you fail to do this, the light won't be pure white light and you'll get some distortions and some halos around your objects. Compound microscopes have two magnifying lenses, the ocular and the objective. And to get the total magnification, you multiply the magnification of the ocular times the magnification of the objective. The ocular doesn't change, and if you'll look on your ocular, you will see that the ocular magnifies 10 times. You can rotate the objectives in the nose piece. This is what's going to change the total magnification that you're getting. Each objective will have a different total magnification. The shortest objective is called the scanning objective, and you will see that it magnifies 4 times, the number right there. So the total magnification of this is 10 for the ocular and 4 for the objective, or 40 times. The next objective is the low power objective, sometimes called the 10x objective because it magnifies 10 times. So the total magnification with this objective is 10 for the ocular, 10 times the objective, or 100 times. The high power or high dry objective magnifies 40 times. And again, look on your particular objective to see the exact magnification power. Sometimes this is 45 or 42. It will vary. The total magnification here is 10 for the ocular times 40 for the objective, or 400 times. The oil immersion lens has to be immersed in oil to collect enough light to get to your eye. It magnifies 100 times, so your total magnification here is 1,000 times. You have to use the oil immersion lens when you're looking at bacteria. When you use your microscope, you want to be able to see things clearly. And two objects can only be seen distinctly as two separate objects if they're far enough apart for light to pass between them. Today's light microscopes are effective at 2 micrometers. That is, things can be as close together as 2 micrometers and you can still see them as distinct objects. That distance between two items, so that you can still see them as two items, is called the resolving power. The resolving power of a microscope depends on the wavelength of the light used and the numerical aperture of the lens. Now, the shortest visible light wavelength is in the blue range, so all of the microscopes have a blue filter over the light source to give you that shortest wavelength light. The numerical aperture of the lens is something that is developed when the lens is made. We have no way to control that. Couple of other terms that are used in microbiology. The field of view, or the field, is simply that lighted circle that you see when you look through the ocular. And then you'll experience something of depth of focus, particularly when you go to the higher magnifications. As you go to the higher magnifications, you will get the sense that there are layers of things, and only one layer will be in focus at a time. This means that typically when you're using oil immersion, you're sort of constantly tweaking your focus to keep things in focus. Because when you turn the coarse adjustment knob, sometimes the stage moves down and other times the objectives move up. It just depends upon your microscope. We're going to refer to this space between the objective and the slide as the working distance. So we'll talk about increasing working distance or decreasing working distance. To start focusing, you want to use your coarse adjustment knob and give yourself maximum working distance. You then obtain a slide and place it on the stage. Now, don't force the slide under anything on the stage. It should just fit into the little holders on the stage. You start with the low power objective. Because we're working with bacteria, the scanning objective is really not very helpful, so we're just going to go ahead and start on low power. Put that in place and use your coarse adjustment knob to get minimum working distance. That is, get the objective and the slide as close together as you can. You want to do this while you're watching from the outside, just in case you have accidentally rotated a different objective in place. If you're jacking that slide closer to the objective and you've got one of the longer objectives in place, you may go right through the slide and break it and also damage the objective. You want to use your stage adjustment knobs to position the slide so that there's something on the slide that's over that hole in the stage. There may be a lot of clear space on your slide. Trying to focus on that won't be helpful at all. Now you're ready to look through the ocular. Using the coarse adjustment knob, begin to move the objective away from the slide. This will start to bring it in focus. Once you get something in focus, you can sort of tweak it with the fine adjustment knob. You're not going to see much on low power, so you're going to want to go to a higher magnification. The scopes we use are parfocal. That means once they're in focus with one objective, they're pretty much in focus with all objectives. So you don't really have to move the stage. All you have to do is rotate the high power objective in place. Don't try to skip this step. Go to the high power objective. You will notice that there is very little working space between the slide and the objective, so you're only going to use the fine adjustment knob to sharpen your focus. You'll get something in focus, but it's still going to be disappointingly small. You're going to have to go to the oil immersion lens. Now, this lens only works properly if it's immersed in a drop of oil, so you've got to put oil on your slide. Do not move the stage. Simply rotate the nose piece so that you're halfway between the high power objective and the oil immersion lens, and put a drop of oil on your slide. Then rotate the oil immersion lens into place. You're going to be afraid that you're going to hit the slide, but you won't. Be sure that the lens is sitting in the oil. If it's not, then you've not focused something correctly. And of course, use only the fine adjustment knob to sharpen the focus. When you're finished viewing that slide, you want to maximize your working distance and remove the slide from the stage. If you want to view another slide, you're going to have to start on low power and go through the entire focusing process again. Glass slides vary enough in thickness that you can't simply slip one out, put some oil on another one, and slip it back in and expect it to be in focus. When you're finished using the microscope for the day, you need to clean the microscope. You start cleaning the microscope by removing the slide from the stage. Then you're going to clean all the lenses with lens paper and, if necessary, some glass cleaner. And since we used oil immersion, glass cleaner is usually something you'll need to use. You start with the cleanest lens, the ocular, and work your way to the dirtiest lens, the oil immersion lens. So you clean your ocular, the low power, the high power, and then the oil immersion lens. Give the oil immersion lens plenty of attention because if oil stays on that lens, it will dry, it will collect dust, and it will make that lens non-functional. Then rotate the shortest or the lowest power objective in place and set the microscope at minimum working distance. Turn off your light, unplug the microscope, coil up the electrical cord and put it where it belongs, and return the microscope to the appropriate space on the shelf.